Okay. Now I can see. So good afternoon, everyone. I think this is the only lecture which is a little bit related to biology. So I tried to connect it to this session by means of energy and devices. So I will try to show you some of our progress related to this issue. But first, I would like to introduce the activity in our group. So we started a molecular scale, or the compound scale. Then we changed the molecule according to the question, to the scientific question that we want to ask, by means of doping, plasmonics, chirality, etc., etc. Then we take this stuff and try to make a self-assembled monolayer or multi-hierarchy self-assembled monolayer, and ask some questions regarding to this. We'll take all this stuff and make devices out of it, and use it to ask some questions related to nanotechnology, material science, etc., etc. Today, I'm going to show and talk a little bit about our devices, our vertical devices, and what we can do with them with respect to bioelectronics. I'm going to show you some examples of natural doping, science-induced doping, and if time permits, I'm going to talk a little bit about light harvesting proteins, and maybe about BioLEDs that we have developed during the last years. So this talk, the basic material will be proteins, which is, of course, biomaterial, which is, from our point of view, is a very nice and very attractive building block in which we can do many things with it, because all of these properties, so it can be water soluble, but may contain hydrophobic moieties. This is very important for us for doping. It can host or release atoms or molecules. It is can be electroactive, photoactive. We can make it self-assembly, etc., etc. With respect to this talk, we are interested to the transport properties of these proteins. We are interested in doping on a nanoscale and in bio-nanocomposites. So first, let's define our tools, which are the molecular junctions. The molecular junctions is basically no more simply saying a molecule or a self-assembled monolayer which is sandwiched between two metals. Okay, we can go into the molecular scale. This is a single molecule experiment in which we have two electrodes and one molecule stuck in between. This is not what we are doing. We are interested in the self-assembled monolayer, or in the films or vertical architecture in which we have a bottom electrode, a top electrode, and a self-assembled monolayer. This is, of course, an ideal picture. We know that it is not the real picture, but this self-assembled monolayer is a sandwich between these two contacts. And then when we have this device, we can measure it by means of current versus voltage characteristics, capacitance, impedance, etc., etc. I will show you just the IV characteristics and try to explain what we can learn out of it. So what we did many years ago was to introduce a reproducible method in which we have a self-assembled monolayer, which is defined between these two contacts. Inside this nanohole, this is the self-assembled monolayer, and the nanodots or the nanoholes method, we could do some kind of this mesh device, some other types of devices. But because this device or this architecture, the molecules are encapsulated, we can build a vertical molecular circuit out of it, because we can build another layer of devices on top of the bottom layer. And here we can introduce one type of molecule. Here we can introduce other type of molecules, and we can build a vertical molecular circuit out of this. So this is nice, but we are and still very interested in the concept of the molecular transistor. And I want to convince you that this is very important in molecular and also in bioelectronics. Why this is important? So a very simplified picture says as follows. Suppose that we have a source and a drain, and some discrete energy levels of the molecule, which is sandwiched between this source and drain. So we apply a voltage, and we have this Fermi window. And all the action takes place between this source and drain with between this voltage span. So this energy level will be in business, but if we introduce some coupled, capacity-coupled electrode, like the gate voltage, the gate electrode, then we can tune the energy levels between the source and the drain and see what is happening in the molecules. So let's summarize why gating is important. We can tune the molecular energy levels. We control, of course, over the potential in the bridge, and we can try to understand what are the limits for miniaturization. So how can we make it? This is technologically very challenging because the gate electrode should be capacitively coupled to a very, very small gap inside the junction. So this is what we do. So this is the junction that I showed you before, but now the size here is not of 100 nanometers. It could be much, much larger. What we did, we introduced a vertical gate electrode inside this hole. This is a metal electrode which we oxidize, and we have a vertical nanotransistor in which the current goes from the source to the drain, and we tune the potential inside this junction by means of this gate electrode. Just to show you, again, this is the source, this is the drain, this is a ring-like structure in which we tune the potential by means of this vertical gate. And this is what we showed, and we published several papers about this technology. So how can we apply it to bioelectronics and molecular electronics, and how can we prove that this works? So let me show you some examples. Before this, for those of you who are interested in fabrication details, this is a fully photolithography and CMOS compatible technique. It means that we can make a sub 10 nanometers channel in a photolithography technique. We don't need any e-beam techniques, and it is CMOS compatible if we use the right molecules, and it got some interest from several microelectronics companies. So these are the transistor characteristics of some protein. This is a bovine serum albumin. This is commonly used in electrochemistry. And you see, and we'll go a little bit into details in few slides, you see that this is very, very sensitive to the gate voltage. This is very important in molecular electronics. It means that the field penetrates very effectively into the gap. So we showed that we can do this. We could self-assemble it by this cysteine moiety, and we proved that this transistor works. So what can we do with this, and how can we understand or try to explore the transport properties of biomolecules? Let me show you some examples. These are IV characteristics that we're taking from several groups that published results on azurine IV characteristics. Azurine is a redox-based protein, and you can see that the IV characteristics that we're taking from several groups show different properties. This is quite disturbing, but not surprising, because each group try to induce its own devices and its own methods, so you see a large mess in the literature. There is a continuing question and still open question: is redox-based proteins like this azurine exhibits NDR as shown here? And does this NDR relates to the redox center? So we thought that we can try to resolve it by our transistors And the logic is as follows. Suppose that we have one group that shows some energy level within this Fermi window in which this NDR relates to it. And the other group, because the condition of the experiments didn't see this NDR peak. So by means of tuning the energy level inside this Fermi window by means of the V-gate, we could compensate and try to see it in different potential inside this energy gap. So this is what we did. And we still don't have the mechanism, but we proved the following. So we ask ourselves, does NDR correlated with redox center? And can we really tune the energy level inside the Fermi window? So first, what we did, we took the apoazurin. The apoazurin is azurin without the redox center. So we scan over the V-gate, and we didn't see any NDR peak inside. Not surprisingly, when we scanned with our vertical gate transistor with the redox center, we see very nice NDR peaks with appropriate gate voltage. It means that the NDR is correlated with the redox center, and we can tune the energy level inside this protein self-assembled monolayer. The mechanism for this phenomenon is still open, and I hope that there are some theoreticians that works on this problem because it's considered very important in bioelectronics. So this is what is called the natural doping, but what we aimed is to do a site-induced or chemically induced doping. If we take this bovine serum albumin, and by means of simple chemistry, introduce one C60 molecules inside one albumin molecule, we wanted to ask ourselves what will be the influence of the doping on the IV characteristics measured by our transistors. So let's go to the results, and I will explain them very briefly. So these are the results plotted in Forer-Nordheim plots. You see that the C60 doped BSA and a BSA show very different characteristics. And from these transistors curves and from other complementary measurement, we concluded that electron transport and governs and not a Hall transport. We can see that we have two distinct transport regimes that we could correlate it after them from Forer-Nordheim tunneling to space charge limited or a trap charge limited mechanism. I will show you in a minute. And you see that the crossing point from at the doped molecules occurs at higher electric fields. One advantage of the transistor is that we can analyze the data as a function of the V-gate. So if you plot these curves, which try to find this alpha, you see that at the doped BSA, alpha is equal approximately to 2. And for the undoped BSA, alpha is not stable. It's going from 5 down to 2.5. With this and with some related spectroscopic techniques, we could find a plausible mechanism which says that there is some bands of defects inside the BSA monolayer. When we doped it, everything is governed by a single trap inside this monolayer. We could identify its energy level, and basically we concluded that we have some kind of a nanocapacitor, which is not surprising. So these are the examples that we have which relates to biology. We have lots of devices that we made with our transistors, memory devices, logic gates, etc., etc. but I want, again, to stick to biology, and I want to show you some results that relates to the Photosystem 1 and what we did with this during the last few years. Again, protein and some relating, some connection to energy. So we're talking about the photosynthesis process in which, by means of light entering this amazing and important process, you have some chemical reaction by means of water splitting and this electrochemical circle. But we were interested in this amazing particle, which is called Photosystem 1. And let's go a little bit to the details, and maybe you will agree with me that this particle is really amazing. So what about this Photosystem 1? So the Photosystem 1 is a light harvesting protein in which you have several parts inside. You have the antenna, you have some kind of reaction center. It absorbed at the visible range at two distinct peaks. We're going to talk about it in a minute. And it has a very nice and very efficient internal quantum efficiency in which we thought that we should explore. So the structure of Photosystem 1 is as follows. So this is a top view. You have a perfect antenna in which light is absorbed. And by means of a process in which is now under debate, this light is transformed to an exciton. And what's important for us is that this exciton is a vectorial exciton, and the voltage drop is around 1 volt over 10 nanometers. So this is amazing. And we wanted to ask ourselves first, is this stuff stable in the solid state? And this work was done with Hanoch Carmeli, Yossi Rosenwachs, Paul Bloom from MPI, and Andreas Hermann from RAD. So first, this is the absorption peaks of the Photosystem 1. You see that it doesn't absorb in green, it absorbs in blue and red. And first thing that Hanoch Carmeli asked himself many years ago, if this is optically active in the solid state phase. So for this, the biochemists produced a genetically engineered cysteine-based Photosystem 1, in which this part can be used for self-assembly on metal surfaces. And if they used two mutants here and here, you can make junctions with it. We also used a surface functionalization with some of our devices, and I'll show you in a minute. So this work was done by Hanoch Carmeli and Yossi Rosenwachs to show that when you self-assemble monolayer and make a self-assembled monolayer of this Photosystem 1, it is optically active. So this is the topography taken by AFM, and this is the surface, the Kelvin probe, the light-induced Kelvin probe that show that you have increase of the contact potential difference when you induce a light. So this stuff is active in the solid state. We showed several years ago, we explored the energy dispersion relationships, and we saw the characteristics of the peaks in the solid state, and showed that it contains also some IR tail, which relates to the dipole between the molecule and the surface. So this stuff is still in the solid state phase, is optically active and alive, and now we ask ourselves what we can do with this. So we did during the years several things with this. It is optically active. It is very stable. So we made some nano junctions, some kind of photovoltaic cell, and recently we are in the business of plasmonic coupling, and I will show you some fresh results relates to this nice stuff. So first thing, we tried to make some junctions with it, and we coupled this Photosystem 1 to carbon nanotubes. We could couple it via one mutant or two mutants to make some junctions via this simple chemistry. And we showed by means of AFM and adsorption that the resulting structure is a combination of the photosystem 1 and the carbon nanotubes. We can make, by using the two mutants, some junctions out of it. The problem with this stuff that we could not control the junctions. So what we did is just measure the IV curves and the photoconductivity as a function of the light intensity. And with this project, we have done. Next, we ask ourselves, can we make some plasmonic coupling of this photosystem 1 to some nanoparticles? We followed a theoretical paper by Sasha Govorov that suggested that this is possible. This is very important, of course, for many reasons, also for solar cells. So these are theoretical calculations. It took us several years and different models suggested by Sasha to show that this is doable. So these are the photosystem 1 pristine structures. These are the photosystem 1 coupled to the nanoparticles. You see that there is nice applications with the silver colloids and the gold colloids as well. Next stage or next step was to ask ourselves, can we extract a power? Can we make a photovoltaic cells out of these structures? So the answer is yes, but I will show you in a minute. So for this project, we used the functionalized surface, and we used the trimer photosystem 1. These are the team images. Hope you can see it, and the self-assembled monoray of the trimers. But when you use this stuff, you have to ask yourself, what is the orientation of the photosystem 1? Is the photo-induced dipoles goes up or down? So for this, we suggested a technique in which we coupled to the photosystem 1 into an active solar cell, and ask ourselves, what is the 5 minutes? Okay. What will be the open circuit voltage if we assume some kind of a dipole here? So if we assume some kind of dipole, our hypothesis was that with a regular solar cell, the open circuit voltage will decrease, and with inverted solar cell, the open circuit voltage will increase. And this is, again, what we got. So for this, when we know the dipole, the photo-induced dipole, we can determine the orientation, and we can make a solar cell in which the photosystem 1 is an active component, the only active component in solar cell. So this is what we did. This is a real biosolar cell in which you can see the dark and the light, and we really extracted the power out of this. So this is a real biosolar cell. And next, we ask ourselves what we can do in order to increase the efficiency of this solar cell, which is extremely low. So we ask ourselves, can we fill this green gap with some other molecules? And this is what we did several months ago. So in collaboration with Andreas Hermann from Groningen, they attached some molecules to this photosystem 1s, and by some kind of threat mechanism, we started to fill the gap between the red and the blue. So these are the results. This is just a reference. This is the photosystem 1. This is the photosystem 1 with the cells, and we really increase the power. But again, the efficiency of this solar cell is extremely, extremely low. It is less than 1%, which is not surprising. It is not surprising because of the green gap, the defects, interface engineering, et cetera, but mainly because of the adsorption cross-section. Okay? There's only one monolayer there, and you cannot, the the light absorption is not efficient. So, of course, you can build many, many monolayers, and this is what was done by Rosenwachs and Nathan-Nelson several years ago. So they used a single crystals made out of a photosystem 1, and the photo voltage is unbelievable. It's of 40, 50 volts. It's amazing, but the current is 0. So the power extracted by this very nice crystal is 0, though the photo voltage is large. So there is, of course, you have to play with this stuff in order to get something out of this. So what we do with this stuff currently, we do a 3-dimensional solar cells, couple it to some nanoparticles, and fill the green gap. And last but not least, before I conclude, these are some new results. These are just slits, okay, empty slits that we covered the walls of the slits with 3 layers of photosystem 1s, and we induced light transmission through these slits. So first observation is that we have a very nice amplification of the transmission. Most of the slits are empty, so this is the dramatic stuff here. And the most important stuff is that you look at with CCD camera on these slits, you see standing waves. Okay, you see standing waves, which are different with respect to the size of the slits, but most of the slits are empty. And these standing waves are directly related to the absorption bands of the photosystem 1. You see that in the red light, we have these standing waves. The green light, we don't. And with the blue light, we have it. So we're still trying to understand this stuff. We simulate it here, but this is in process. And this I will not talk about. Sorry. And I want to thank you for your attention.
Time for a few questions. Yeah, Mark.
When you see the NDR,
yeah,
and you reverse the voltage sweep, is there hysteresis?
So it depends on what type of molecules. With this protein, we don't have hysteresis. With other types of molecules, for example, ferrocene, we see some hysteresis.
How about the sweep rate? Is there any dependence on sweep rate?
Yeah. So in these molecules we didn't check. There are molecules that we see some sweep. We see some depends in the sweep rate, which relates to, of course, charging. Yeah, we see it.
Any other questions? Okay, let's thank Shachar.